transmembrane protein is a type of membrane protein spanning the entirety of the biological membrane to which it is permanently attached. That is, transmembrane proteins span from one side of a membrane through to the other side of the membrane. Firm attachment of TP to biological membrane is aided by a special class of membrane lipids, called annular lipid shell. Many TPs function as gateways or loading docks to deny or permit the transport of specific substances across the biological membrane, to get into the cell, or out of the cell as in the case of waste byproducts. As a response to the shape of certain molecules, these freight-handling TPs may have special ways of folding up or bending that will move a substance through the biological membrane. Transmembrane proteins are polytopic proteins that aggregate and precipitate in water. They require detergents or nonpolar solvents for extraction, although some of them can be also extracted using denaturing agents. All transmembrane proteins are integral membrane proteins, but not all IMPs are transmembrane proteins. Types. Classification by structure. There are two basic types of transmembrane proteins, alpha helical and beta barrels. Alpha helical proteins are present in the inner membranes of bacterial cells or the plasma membrane of eukaryotes, and sometimes in the outer membranes. This is the major category of transmembrane proteins. In humans, 27% of all proteins have been estimated to be alpha helical membrane proteins. Beta barrel proteins are so far found only in outer membranes of gram-negative bacteria, cell wall of gram-positive bacteria, and outer membranes of mitochondria and chloroplasts. All beta barrel transmembrane proteins have simplest up and down topology, which may reflect their common evolutionary origin and similar folding mechanism. Classification by topology. This classification refers to the position of the N and C terminal domains. Types I, II, and III are single-pass molecules, while Type IV are multiple-pass molecules. Type I transmembrane proteins are anchored to the lipid membrane with a stop transfer anchor sequence and have their N-terminal domains targeted to the ER lumen during synthesis. Type II and III are anchored with a signal anchor sequence, with Type II being targeted to the ER lumen with its C-terminal domain, while Type III have their N-terminal domains targeted to the ER lumen. Type IV is subdivided into 4A, with their N-terminal domains targeted to the cytosol, and 4B, with an N-terminal domain targeted to the lumen. The implications for the division in the four types are especially manifest at the time of translocation and ER-bound translation, when the protein has to be passed through the ER membrane in a direction dependent on the type. Thermodynamic stability and folding. Stability of I plus or minus helical transmembrane proteins. Transmembrane I plus or minus helical proteins are unusually stable judging from thermal denaturation studies, because they do not unfold completely within the membranes. On the other hand, these proteins easily misfold, due to non-native aggregation in membranes, transition to the molten globule states, formation of non-native disulfide bonds, or unfolding of peripheral regions and non-regular loops that are locally less stable. It is also important to properly define the unfolded state. The unfolded state of membrane proteins in detergent micelles is different from that in the thermal denaturation experiments. This state represents a combination of folded hydrophobic I plus or minus helices and partially unfolded segments covered by the detergent. For example, the unfolded bacteriorhodopsin in SDS micelles has 4 transmembrane I plus or minus helices folded, while the rest of the protein is situated at the micelle-water interface and can adopt different types of non-native amphiphilic structures. Free energy differences between such detergent denatured and native states are similar to stabilities of water-soluble proteins.